will start to explaining this research. Before, I want to bring some brief explanation about this project and the place that this project was done and the place that I am here. It's a comparison the immunogenicity of HIV-1 P24-Nef protein. It's a peptide vaccine conjugated with two adjuvants. One of them is ISA, Montanide ISA 70, and the other one is FliC protein. Reserved. It is produced from pseudomonas aeruginosa pathogen agent. In BALB mouse model in 3 different injection immunization. This project was done in Tehran Institute Pasteur of Iran. The group that works, they are working on HIV vaccine and peptide designing as a vaccine. Through that days that I was a research assistant, I did this project. After that, I turned my way from HIV research to cancer research, and especially in clinical base, and changed my place to the hospital, Hospital of Mashhad. And ultimately, I am postgraduate student of University of Manchester here. I received this peptide vaccine from Dr. Mahdavi, who published several articles, and he spent his time on HIV vaccine designing HIV vaccine. And I received the FliC protein for this project from Sobhan Faizi, who parallelly on that time works on making recombinant FliC protein for patients who suffered from a wound of burning, heavily burned patients in the hospital. And after that, let's start with introduction to this project. Still, HIV is a problem for human being and for human's life, and scientists, researchers try hard to find a way to introduce or find an effective vaccine with effective response, both for humoral and cellular responses. For this purpose, vaccines HIV vaccine needs new strategies for making peptide vaccines or DNA vaccines and enhancing these responses by adjuvants. I bring a whole genome of HIV just to show that 2 parts of 2 protein of all protein that HIV has had more important role in peptide designing as a vaccine, because they have less variation and variety, and they have through several research it shows that it has more effective response for immune response. It's one of them is a GAG protein. It's a protein of nuclei. And it's one part of that is P24 in our peptide that we used. And the other one is Nef protein. It's a regulator agent, very small protein, and very effective, brings effective and conserve immunogenicity against HIV. In fact, all these matters brought together to Nef as an important target of immune responses in HIV-infected people, and it is recognized by the immune system which responds to it. Then, in recent years, it is needed to enhance and boosting the immune response with more effective response that using adjuvants. There are 2 types of adjuvants. One type is adjuvants, delivery adjuvants, that mostly they are a combination of oil and water that just deliver the vaccine to the cells or to the part that they needed. The other type of adjuvant is the adjuvant helps to inducing a robust and effective response and promote a strong adaptive response. For these two matters, we used two adjuvants. One of them, as I said, from pseudomonas aeruginosa agent. That's an opportunistic pathogen invade patients with a lack of immune system, and they have immunodeficiency, like cancer patients or HIV positive patients, or people with very heavily burned wounds. This pathogen has got flagellin. That the main protein is FliC. FliC, it was revealed that FliC has an important role in have a connection with TLR5, tool-like receptor, especially number 5. This tool-like receptor can brings and induce the inflammatory response and can stimulate innate immune responses. Then, by considering the connection of stimulating innate immune response with the other responses, that will be a good adjuvant for us. But we guess that. So all of these matters brings to the aim of this project design, that we find that an explanation on the possibility of using flagellin in combination with Montanide ISA 70 as a candidate vaccine, and in that peptide that found that HIV-1 P24-Nef is most effective peptide through several peptides that he has found, and using and trying different ways of immunization and injection, intramuscular, subcutaneous, and intradermal ways of injection, because it is known that the way and the route of injection and immunization brings different responses, especially for T helper 2 and T helper 1 or memory cells. Okay. After this, I bring a brief method. We received vaccine and FliC recombinant protein, grouped mice and immunized them in 0, 14, and 21st days of their working, gathered the blood serum and spleen cells and cultured these cells. Then the immuno test, lymphocyte proliferation assay, was done through BRDU kit, and interleukin 4 and interferon gamma ELISA assays and ELISPAS test was done in order to assess both cellular and humoral immune responses. About the groups that we have, we had three different groups except our control groups. One group, HIV-1 P24-Nef FliC with both adjuvants, FliC and ISA Montanide. The other one just in 3 different routes: subcutaneous injection, intradermal injection, and intramuscular injection. Then HIV-1 P24-Nef with one adjuvant, Montanide ISA 70, again through 3 different injections. And the last, FliC in Montanide without our peptide, because we want to know about the application of our peptide and whether it is useful or not. About the dose that we use, It was 20 microgram per 100 microliter of injection in every time in 3 different injection. As I said, 0, 14, and 20 first day of the we start work. And beside these groups, we had controls as adjuvants with PBS and the PBS with itself. Okay. All the procedures after 3 injections on the 42nd day, we gathered blood sample and collected candidate punctures. Cardiac punctures and the splenal cells. Then it takes after its final boosting, lymphocytes proliferations and interleukin-4, interferon gamma through ELISA tests were done, and lymphocyte proliferation with BRDU, as I said. It's some pictures from that days the injections of mice and spleen separations and their cells culturing and cells gathering in the plates of the cells, and the ELISA test for these procedures. Okay, what about our result? Through all results, we have very significant difference in our group 1, the peptide with both adjuvants through subcutaneous injection, except for antibodies, because these are completely different for antibodies, these results. As you can see that they are in the first group, for lymphocyte cell proliferation in BRDU, we have more index. Again, for cytokine interferon gamma, in the first group, we have completely effective response. However, we don't have any special or effective response for the last group, FliC and Montanide, without peptide. For interleukin-4 again we have this, and for interferon gamma ELISpot test we have these differences with other groups in subcutaneous way. So it was revealed that subcutaneous way with our both adjuvants and peptides completely effective for us. And I brought ELISpot test picture that shows that completely spots that in positive group and in P24-Nef FliC group and P24-Nef, and the difference of the presence of these spots on ELISpots. About total antibody, but for total antibody level, we have the most effective response in intramuscular injection immunization. It is different from others, that it brings our conclusion that for antibodies, having more antibodies, intramuscular injection may be better, but it needs more work to receiving a conclusive conclusion. But as you see, for total antibody level in group 3, we have a higher response, especially when I go back to the intramuscular responses. You can see that better responses in these matters. What's our conclusion? Unfortunately, it was the first, maybe fortunately, it was the first time that the FliC recombinant, new recombination of this FliC was used for peptide vaccines. That is really indeed in need for having test projects on peptide vaccines for HIV. Besides some adjuvants like FliC that is completely shown that this molecule has a effective response both for humoral and cellular responses. And the route of vaccination through FliC adjuvants show us meaningful responses. Okay, I have a question.
Thank you, Arezoo.
Thank you for your time.
Okay, do we have any questions for Arezoo? I was wondering what, in terms of your measurement of response, what would be your goal? What would be indicative to you of a successful candidate? What values, or where do you stand with respect to that?
This project is a little part of a whole project. It as illuminating the way of other researchers or students in that institute. It was done. Through this, it was found that for further investigation, they should use subcutaneous injection with more robusting or injection, or maybe with changing the peptides in effective way or stuff like that. I don't know I can make a response to your question or not.
A related question for me is, we've seen so many attempts at an effective vaccine. Do you really think it's possible? Is there, using these classical approaches and choosing different antigenic moieties, do you think it's possible?
Everything is possible in the world as a whole, but it really needs time. I don't know, going to HIV path is very difficult, and it's you don't know what's the next step. You don't know, is it effective? Am I working and trying hard, bring some results for people in clinics? Because of that, myself changed my way to cancer research. I'm pessimistic about.
I've become that way too. They're certainly gaining a lot of information. You're learning. You're presenting some interesting results to improve the general field, but it's very frustrating.
Yes, it's like a hallucination. So
any other questions?